All the individuals in the population were little a. And then along this lineage, let's say you had no mutations occur in the a at this locus, or if they did occur, that they did not spread through the population. So all the individuals in the species on the left, we'll call the left-handed species, all have little a. Now, in the right species, let's imagine a case where we have a big A mutation occur. So when that mutation occurs, when the big A mutation first occurs, it lands on one chromosome in one individual, right? And then later it can spread through the population. So let's imagine that by this point, all the individuals are big A, big A. That is to say that big A mutation later became fixed in the population. There's a period during which the population was polymorphic for the big A allele. There's some individuals, the population had both big A and little a, but now it's all big A. So the wrong model goes like this, that when you have hybrids form, well, they have to all be heterozygous, right? No matter what. This is just Mendelian genetics. When you have a little a individual mating with a big A individual, the offspring that's formed is heterozygous. It has to be. So the wrong model, and people argue this for a while, for not very long, is a model where there's something wrong with the heterozygous case. Right? For whatever reasons, the heterozygous individuals aren't as fit. Right? So these guys have a lower fitness. Well, what's wrong with this model? Why is this not a good model for describing what types of genetic incompatibilities are causing hybrid sterility or death? Anybody see the critical flaw? Well, go ahead. You need heterozygous individuals to get that. That's exactly right. So if these guys are bad, then so are these guys, right? And so you'd have to go through an adaptive valley to get all A, big A, big A. So if these guys are bad, then these guys are bad, and it's unlikely that natural selection or even genetic drift would get you through this step right here. That's the wrong model, and that's why it's wrong. Right? Now let's go to what we think is a good model. Model that's actually supported by evidence, the data. Not only is it a sensible model to write down on a board, but there's more and more data accumulating that this is a model that looks like it's actually in action, that actually works. So this is a two locus model, though in principle it could apply to many more than two loci. And before I get there, let's just review something very briefly. I mean, briefly like it's in a minute. But here we have DNA, right? There's parts of the DNA that we think are that code for genes, for proteins. Basically, you have messenger RNA, which is basically this region of the DNA is transcribed into messenger RNA. Then this messenger RNA can be post-processed and through a process called translation, read into proteins. And these proteins themselves can fold up on themselves and form complicated structures. And in fact, it's the folded protein that does all the stuff in the body. So these folded proteins can perform structural function in the cell, or they can be used to catalyze reactions and so forth. So all the work in the cell is done by proteins. Okay, so this is something you get in 1A if you haven't already gotten it. But I want to point out something, and that is to say that most structures, the proteins very rarely work in isolation. Okay? It's almost always the case that proteins have to interact with other proteins. So to say, proteins that come from other parts that are transcribed and translated from other parts of the genome. So you might have a protein here that has to work with, this is like the first time ever where it would be nice to have a different colored chalk, but this is fine. They have to interact with another protein, or maybe even more. But this would be an example of protein 1 and protein 2, where then for the function to be performed, both of these proteins have to interact one with the other. Right? That is the typical case in the cell. Right, so that's background. You may have already known that. If you didn't, well, there it is. So this is the two-locus model where you have one locus for protein 1, another locus for protein 2. We're going to call them the little a, or the A locus and the B locus. So let's imagine a case where ancestrally we're little a, and little b, little b. And that's our ancestral condition. Okay? And along this lineage, we'll imagine that there was a mutation in the A locus, so that at one point we had individuals that were little a, big A, and little b, and that by the time you get up here to the present, that this big A mutation or allele becomes fixed in the population. So all individuals over here, big A, little b. Well, imagine the opposite occurs over here, that we have little a, that we have a mutation in the B locus, such that by the time we get over here, all the individuals are little a, big B, big B. All right? So that is the situation. I just made up, but this is a model. It's a simplification of reality. So the hybrids that are formed, well, they all have to be little a, big B. Okay? They all have to be that sort. And the important point is this: these hybrids are unique in one special way, and that is that the big A locus, the big A allele and the big B allele, they have never ever in their entire evolutionary history seen each other. Right? You see that? This is the first time in the hybrids, the big A and the big B alleles. This is the very first time they ever see each other. Right? And there's no guarantee that they will work together, like they might have to. They might have to interact in some way. There's no guarantee that they'll work properly together because they've never been tested together. That's, when the big A allele arises, it was only ever forced to work with little b variants of the protein, right? And similarly, when the big B allele arose, it only ever had to work with the little a version of the protein. Big B was never forced to work with big A, and there's no guarantee that they will work the very first time they see each other. So the idea is that incompatibilities between these loci that uniquely arose on these different lineages, incompatibilities in the hybrids is what causes a lot of the failures in development, or causes a lot of post-zygotic isolation that's out there. This model has a name. It's called the Dobzhansky-Muller model. Or the Dobzhansky-Muller model. It's a model for describing what types of incompatibilities could be arising that are causing reproductive incompatibility between different species. Why do the hybrids not work? Because of incompatibilities like this. Now, it turns out that these are very difficult to study, but there's been a lot of progress on uncovering examples of Dobzhansky-Muller model incompatibility. The Dobzhansky-Muller incompatibilities is what they're called in some model organisms. So there are people that study, a lot of evolutionary biologists study fruit flies, and there's been a lot of examples of these types of incompatibilities uncovered between crosses between different species of fruit flies. Okay? If you're having trouble seeing it, there it is. All right. Is there any questions about that model? Now, you might ask, well, how many Dobzhansky-Muller incompatibilities do you need to arise before you have incompatibilities arise between species? That is to say, before the hybrids, for the hybrids to work, and it may vary. It could be that you need hundreds of such incompatibilities to arise in order to have hybrid sterility or have the hybrids that are formed die. Okay, be legal. There's a couple dozen examples of this now, but there's probably hundreds of them or thousands of them that could be uncovered for any particular cross between species. Okay, what do we want to do next? Okay, so that is what I wanted to say about the types of differences that can rise after you've had gene flow turned off. Right? You have the pre-zygotic and post-zygotic isolating mechanisms that can arise over time that cause two species, when they come back into contact, not to form successful hybrids. Right? If the time has been sufficient for many of these incompatibilities to arise, that when the species do finally come back into contact, it just doesn't work. They don't recognize each other as being good species, individuals from other species as being individuals they can mate with. There can be mechanical means that even if they wanted to, it wouldn't work. Or if they do happen to mate, there's a large number of reasons why genetic reasons why the hybrids that are formed will not work. Let's go now to what are the initial mechanisms by which you can turn off gene flow. Remember, the reason why you have these populations diverge in the first place is because mutations are accumulating independently in the two different populations. And there's two different types of, a lot of this lecture is coming in twos, it turns out. The first type of isolating mechanism that can occur, that the mechanism that will turn off gene flow, is called allopatric speciation. And allopatric speciation is when some sort of geographic barrier prevents individuals from migrating from one population to the next. Okay? Now these types of barriers can be of various sorts, and I would imagine that any particular speciation event, the specifics are going to be different. But in general, you can have what are called vicariance events. These are types of barriers which physically separate two populations, a stream, a mountain range, whatever you might imagine, a desert forming in the middle of a population. The other type of event is called a founder event. This is a case where you have a population of individuals. We talked about this with the founder effect in population genetics. When you have a small population being founded some distance away from the main population by a small number of individuals, but the point is that this is a very rare event. This founding event is something that would be expected to occur only very infrequently. Then you can essentially have very little or no gene flow between this new population and the parental population. Okay? Think volcanic island as an example. Okay? Let's go ahead and I think I've got some examples here. So here's an example of a volcanic island. This is the Pacific Ocean, and we have the Hawaiian Islands there, right here. The Hawaiian Islands are volcanic in nature. They're formed from the volcanic action. And interestingly, as you go further north and to the west, the islands get progressively older and older. So for instance, Kauai, which is near the tip of this range of islands, is the oldest and much older than the main island of Hawaii, where you have active volcanism. Okay? And if you go south and east of the main island, there's no island there. At least there's no island there yet. There's actually an undersea mountain that is slowly growing and growing, and eventually the idea is that eventually there'll be another island formed to the south and east of the main island of Hawaii. The reason why we think that's the case is there's some sort of hotspot in the magma below, and the plate, we haven't really talked about plate tectonics. How many people here have heard of plate tectonics? Oh, my goodness, all right. So the idea is that the plate is moving, and it's moving over a stationary hotspot, and as it moves over the hotspot, you have new island form, and it keeps moving, and new island forms, and so forth. So that's what's the idea. But the point here is that at some point you have a brand new, fresh, very rocky island form that can be colonized. Okay? And a lot of the colonization that occurs now is probably is from other Hawaiian islands, because they're the closest bodies of land. But when you have these colonization events occur, the idea is it looks something like this founder event type model. Okay, you have new bird species, new insect species arises on an island, and because it's a fairly good distance between islands, you can have speciation form. You have very little gene flow, cessation of gene flow between the parental population and the new population on the new island. So what you often see in a lot of these island species is radiations into of specific groups. So here is a radiation of birds that occurred on the Hawaiian Islands, honey creepers. Okay? There's also been a magnificent radiation of fruit flies. I mean, one of the biggest radiations of fruit flies is actually on the Hawaiian Islands. There's a lot of interest among evolutionary biologists to study the fruit flies on those islands. Here's an example I wanted to give as well. This is not of a founder event, but more of a vicariance event. So these are a type of shrimp that live in the Pacific and also in the Atlantic Oceans. So they live on both sides of the Isthmus of Panama. Everybody here has heard of the Panama Canal, right? So let me just give you an idea of what's going on, what we think is going on here. Thanks. So here's a very, I'm bad at maps. So here's North America. Here is South America. And then here's Central America, right? And, well, it's good enough. And there's the Panama Canal. This is the Isthmus of Panama. And of course, the point is, if you really, really wanted to, I wouldn't recommend it, but you could in principle walk from San Francisco to the southern tip of South America, right? It'd be a long, grueling trip. There'd be areas you probably want to be careful in, but in principle, you could do it. But if you went back 3 million years ago, the trip would be complicated by the fact that the isthmus here there was a strait, water. Right? The Isthmus of Panama was below water 3 million years ago. Which means that if you were a population of shrimp or any other marine species that had a range both on the Pacific side and the Atlantic side, you could have gene flow if you were from the Atlantic to the Pacific side. Right? When the Isthmus of Panama rose up about 3 million years ago, all such populations were cut in half. Right? This is a vicariance event. And so there's many examples of what they call gem-knit species. But basically, if you ask what is the closest species of the shrimp on the Pacific side, well, its closest relative is on the Atlantic side. There's many such pairs of species on both sides of the Atlantic where the closest relatives are on the Atlantic side of the isthmus, and vice versa. Okay? So this is an example of what we think is a vicariance event, allopatric speciation through vicariance. And the interesting thing is you can actually date when the gene flow must have been turned off 3 million years ago. And many of these types of species these types of gem-knit pairs have been studied many times. But here's an example from 1993 where Knowlton et al. Looked at divergence in the snapping shrimp and found all sorts of divergence in the proteins, the mitochondrial DNA, and there was reproductive isolation in the reproductive compatibility between the two species on both sides of the isthmus. Right? But here'd be an example we think of a vicariance event. Okay. Going back a little bit too, I've got some other examples. This is going back to mechanical isolation, so pre-zygotic isolating mechanism. Here's examples if you want of sort of fly corn, I suppose. But these are the genitalia of the males in different species of Drosophila. Okay, and the point here being that these are very rapidly diverging part of their anatomy, and some of these genitalia just will not fit into the females of another species. How am I doing on time? So this example I want to give is going to take more than 3 minutes. So what I'm going to do is break here in the first 5 or 10 minutes of the next lecture, finish up speciation, and we'll go to phylogenetics.